I will report on the work that is performed in the lab Minou Russell Zadegan, mostly by Minou itself actually, and search for experimental mouse models of non-Mendelian heredity. Is that, yeah. The criteria one wants for such models are genetic homogeneity and stability of the genomes, which is something which cannot be attained in human studies because of the enormous heterogeneity of our genomes. Of course, clearly defined phenotypes, and, if possible, paternal heredity. Paternal contribution in the mouse can be limited to the very short time necessary for the sperm cell to reach the oocyte. And we have, over the past years, described three models of epigenetic variations which were transmitted in a non-Mendelian way, which affect the expression, it's upregulation, in fact, increased expression of three genes, KIT, giving a very easily recognizable four-color phenotype, which is convenient for the geneticists, CDK9, the key gene in the growth of the heart, embryonic heart, and the overexpression leads to cardiac hypertrophy, and SOX9, which is active in early and late stem cells. Increased number of stem cells in the embryo leads to increased body size, the giant mouse, which interestingly, but that I cannot tell you much than the basic result, is improved cognitive ability. It's very clear that the behavior of this mouse, it is much more intelligent, so to speak, than the average mouse. Trials are in progress currently, and I cannot say much more. In all these three models, we have, as I just mentioned, upregulation at transcriptional level of a key regulatory gene. The modified phenotype is inherited up to 3 generations in outbreeding. I mean, if you breed the modified animal with a wild type, the progeny will transfer, and so for 3 generations. In inbred crosses between brother sister or between the paramutants, it is apparently maintained continuously. We cannot say for how long, how many generations exactly, because with the mouse you have only 3 to 4 generations a year. So when we search for the transgenerational signal initially, we were thinking of cytosine methylation or chromatin structure, and that is probably obvious after this day of meeting. Since we had no indication of neither one of these characteristics were modified in the paramutant, we looked for a possibility of transfer by the sperm of non-coding RNA. And she, Minou, developed the technology of microinjecting RNA into the one-cell embryo, which is very well supported by the embryo. After implantation in foster mothers, we have a good recovery of mice who, if you inject the sperm RNA or the somatic RNA of one of the variants, all the litters show the modified phenotype, and they do transfer in our hands. I mentioned that because a recent paper from an American group agreed essentially with our result, except that in their hands the RNA transfer don't transfer into. We have to check what's the differences, but in our hands it's clear that we have we establish the heredity. The inducers are RNAs with sequence homology to the locus, including the cognate microRNAs. We had problems to further study the molecular level what could happen, because in the one-cell embryo is not the best system to make molecular biology. And now we are in a better shape because we have found that we can see the same increase in transcription in transfected ES cells, embryonic stem cells, transfected with the same RNAs. And the first result at this stage indicate that it requires establishment of the upregulation of the transcription requires transcript of the complementary strand to be present, and also, not mentioned here, the RNA machinery. So this is just a brief summary of several years of current work, and I will just now speak about two projects currently in progress, recently started. One is the paternal heredity of an acquired pathology, and as we heard from Dr. Lillenbeck, eating too much leads to a lot of problems, including obesity and diabetes, and there are indications that these phenotypes are inherited, but at least in the mouse we can set up conditions so that we think we don't have at least cultural or other interactions with the progenies. And what we did so far just show first series of experiments in which we feed the fathers, males, with a high-fat diet. It's 50% butter. The mouse loves it and eats a lot and becomes obese and diabetic. And if these males are mated with a normal female and grown with a normal diet, and immediately after mating, the female continues on a normal diet, and during all the time until the young are grown, and then we see that all these the babies, males or females, have a body weight much higher than the controls. So it's clearly inherited in this sense, and now what we did was to take RNA from the sperm. This is sperm RNA from the initial obese male, which has been microinjected into embryos. You can see that sperm RNA is able to transfer the obese phenotype to the progeny. The next slide will just in fact shows that by the classical glucose response, insulin response test, that these mice and the progenies are not only obese but also diabetic. Now I will turn to a different subject, a different family of diseases, in fact, which are family of the which are diseases of the aging, which are associated with the shortening of chromosomes, shortening of telomeres at the end of our chromosomes. Just to remind you about the basic of when our chromosomes have to be replicated at every cell division, and the replication machinery is functioning in such a way that a double-stranded linear molecule cannot be replicated to its very end. It always will remain at the end 1 to 200 or few hundred nucleotides of single-stranded DNA, which then will be recognized by the cellular surveillance systems, will lead to nuclease attack, to shortening of the chromosomes. And in fact, this is prevented, at least for a long time, for a number of cell duplication, by the presence of a long stretch, telomere, a long stretch of non-coding DNA at the end of every end of our chromosomes, with this characteristic repeats in human and mice, at least, TTA-GGG for several hundred and even thousand of nucleotides, double-stranded DNA. This is done by associated complex association of proteins. Important one is called telomeres. It's reverse transcriptase. It's the enzyme which synthesizes the repeats, using as a template a sequence which is provided by an RNA molecule which is associated with the protein. Okay, we can forget about all the others. It's becoming more and more complex when you get into that, but those are so far the basis. Now, there is one more element which has been recognized relatively late, a few years ago. It's a long RNA called TERRA for telomere, something associated RNA, and it's a transcript made by polymerase II of one of the strands of starting from a promoter in the in the subtelomeric, in the cellular subtelomeric sequences. And this TERRA RNA is, as the repeat of DNA itself, is quite variable in length, but can be very long, up to several thousand nucleotides. The recently published results, especially by Azarin in Zurich, and our own current results that I don't have time to explain here, suggest that the RNA is engaged at the end of the chromosome in an what's called R-loop complexes, meaning that the RNA, one of the strands of the DNA is displaced by the RNA, and there is a long RNA DNA hybrid, which can be identified, for instance, by antibodies which are specific of this structure. Okay, so those are the basis. Now, why were you interested in telomere biology? One point is that the length of telomere, there are several studies, only two are indicated here, but there are several more, which show that the length of telomeres in humans, which is quite variable, but when extensive studies repeatedly shown that the telomere length is inherited from the father. And so that's one reason to be, for us, that was one reason to be interested. The other reason that being that 2-3 years ago now, we made in collaboration with the RIKEN at Yokohama large-scale sequencing of the mouse sperm RNA, and we have evidence this TTA-GGG repeats. We have evidence that TERRA is present in the sperm RNA. And the laboratory of Kravets, who initially first described RNA in human sperm and sequenced it later, they found also these TERRA molecules. So is there a relationship between the presence of TERRA RNA in sperm and the fact that the length of the telomere is fixed by these the sperm molecule? So another element to before getting the results is how is telomere length established in human and in mouse. At fertilization, the telomeres of the two pronuclei are very short, and they are growing during the pre-implantation stage. At least as soon as transcription is started at the 2-cell stage, telomeres is made, and finally the size of the telomere is fixed at the blastocyst stage, at the time of implantation. And in most cells it will not grow. They will not grow anymore. In somatic cells, as indicated here, they will decrease with age. They will be maintained by maintenance of telomerase synthesis only in stem cells. And they will start again in cancer cells, actually. But essentially, everything is done between the cleavage stage and the blastocyst. So what happens when we microinject? We have here a series of blastocysts, of groups of blastocysts. In fact, each bar is for present 5 embryos which have been microinjected with these oligonucleotides, TEL3, TEL2, which are different lengths of the TTA-GGG repeat. And the last one on the right is the control. And clearly, all of them have, all of the injected ones have increased length of telomeres. And this can be also repeated if we transfect the TERRA RNA in embryonic stem cells, once again providing, we hope, a better system to further study the... so it's about where we stand right now. And what are the possible interpretations that we can figure out? We can think of induction by TERRA RNA of telomerase transcription in similar way to the increase in transcription that we noted in the paramutants. And first series of experiments, here individual groups of 10 blastocysts in 3 experiments. That's a lot of mice, I can tell you, to just one graph like that, but it clearly shows an increase in the RNA encoding the telomeres. It's the telomerase RNA which is measured here. Okay. The other possibilities that we like to consider is derived from a very recent paper from the laboratory of Pascal Chantran in Quebec, which shows that the TERRA RNA create a structure at the end of the chromosome which will nucleate the telomeres. So the two models are not exclusive from each other, actually. And what we like to conclude at this stage is that we have good preliminary result to think that TERRA RNA is one more example of long-term non-coding RNA, which is crucial for the regulation of this biological process. Okay, thank you for your attention.
Questions? Oui, Geneviève?
I was actually wondering whether in the experiment where you have injected the TERRA RNA, if you have looked by fish, if you change the pattern in terms of the staining that you can detect in the embryo in light of your hypothesis with the nucleation. So do you have more, or is there any particular visible effect at that level?
That has clearly to be done. Once again, it's still, results are still preliminary, but it would be interesting and necessary to do this type of assay, I agree.
Because by fish, when we looked at TERRA RNA in the early embryo, we didn't see much of a difference.
We are collecting the tools to do that. Yes. We would be glad to talk further if you're interested at some point.
François, maybe I missed the point, but in the striking RNA transfer experiments in the obesity model, how did you, what was the RNA that you used? Because when we look back at the previous talks, there are many possible genes involved in the obesity transmission. So what was the non-coding RNA that you used for transfer in the obesity, in the first experiments?
And heredity of the
overfeeding.
obesity.
Yes.
Okay. No,
can you speak close to your microphone because sorry.
I showed about the results what we have. We injected comparison either testis RNA or whole sperm RNA, and the next question we ask and are still currently asking is that since the control of RNA of the sperm of the normal mouse had no effect, was to compare by sequencing these two RNA preparations. And the answer is quite complex, in fact, because there are no, we didn't as yet found any signal for one or a few candidates. We have several whole series of small up or down variations in different RNAs, and that's about what we know.
Last question by Giacomo Cavalli.
Yeah, François, I was wondering, because it seems to be a quite widespread phenomenon. In fact, you can induce a lot of different phenomena with injection of RNAs, and I wonder whether you have found some cases in which it doesn't work, like if you inject RNAs that don't induce a phenotype. Because I don't recall what has been done, like for instance with microRNAs.
My translator here, because I have some, sorry, I have some hearing problems, so that's just.
Are there any RNAs that you've injected that don't induce a phenotype?
Any RNA
that you've injected into early embryos that do not lead to a phenotype?
We have always controls in which in every
I'm sure.
Every injection experiment.
But for instance,
okay.
So you have tried several microRNAs.
MicroRNA.
And they induce phenotypes. But do you have some that induce and others that don't?
The experience we have with microRNAs are what we published. The microRNA is specific of the KIT RNA, and these two are very efficient to induce. And then we tested a series of microRNA with variable responses. Some, one of them, I think it's mu319, if I'm correct, produces obese, once again, mice, and another one, but most of them had no clear answer, except one, which is MIR124, that we expected to have an effect on the growth of the kidney, and which showed the growth of the whole body, and we believe now maybe growth of the brain also. So that's about what we know. It's just a large amount of work. So
yeah.
we cannot have probably all the controls, and we need a small factory for this.
Thank you.
Thank you.